We are on part 3 of the chapter 3 lecture notes, and we are still covering passive transport processes. So no ATP is required for these passive transport processes. We had learned about simple diffusion, which is just when molecules that are lipid soluble and small and not water soluble, those molecules can simply diffuse from their high to their low concentration through the phospholipid bilayer. But some molecules, which also need to move across the membrane by diffusion, are not soluble in lipids. They are soluble in water. So, in other words, molecules that are polar covalent molecules, like water, or else they are ions with a charge, but they are soluble in water, not lipids. These molecules have to move through a channel or a carrier protein. So, facilitated diffusion is a transport process carried out by carrier and channel proteins, but there is no requirement of ATP. The substances that pass the membrane this way are going to be large, water-soluble molecules, or they're going to be charged. They're going to be ions. Amino acids will move this way across the membrane. Glucose will move this way across the membrane. Sodium ions or any ions. And this is how a carrier protein works in facilitated diffusion. Glucose, as you can see on the outside of this cell, is higher in concentration than it is in the cytosol inside the cell. So glucose is wanting to move from its high to its low concentration. So it does this by moving through a carrier protein into the cell, and there is no requirement of ATP. Now, in active transport, rather than passive transport, there is a requirement of ATP. ATP is required. And now we see substances moving against their concentration gradients. In other words, toward their high concentration, okay? Against their concentration gradients, or from low to high, toward their high concentration. And it allows the substance to accumulate either inside or outside the cell on one side of the plasma membrane. So, the example that we are going to look at is the sodium-potassium pump. The active transport of this sodium-potassium pump is, and notice it is a counter-transport pump. It is an antiporter. So what we're seeing is in step 1, sodium is being pumped outside the cell. What you don't see is sodium is already high outside the cell. Sodium is being moved toward its high concentration. And then ATP is going to bind to this pump and be broken down to generate the energy required to pump potassium. So 3 sodium ions are moved out of the cell and 2 potassium ions are moved into the cell. And what this does is it concentrates sodium on the outside of the cell and it concentrates potassium on the inside of the cell. And it creates a voltage across the membrane, or we call it a membrane potential. It creates an electrical charge across the plasma membrane. And that is necessary for muscle contractions and nerve impulses. So make sure that you know that 3 sodium ions are pumped out. 3 sodium ions are pumped into the ECF, or extracellular fluid, and 2 potassium ions are going to be pumped into the cytoplasm. And I'm going to skip secondary active transport, but it uses the energy, the concentration gradients created by primary active transport to generate the energy to do more active transport. And it's actually really interesting, but this chapter is very long. If you haven't noticed, it's got a lot of material in it, so we really can't cover everything without it taking forever and ever and ever. So vesicular transport or bulk transports a large amount or bulk amount of a substance, either a fluid or a solid, into the cell or out of the cell. So endocytosis is movement of a large amount of material into the cell. It can be phagocytosis or cell eating. That's when a solid particle is ingested through the formation of a large vesicle. In penocytosis, dissolved molecules or solutes and their solvent, so basically a solution is ingested and a vesicle forms around a solution. So that's called we sometimes call that cell drinking. Phagocytosis is cell eating, penocytosis is cell drinking, and then the overall movement of a substance out of the cell, a large amount of substance out of the cell through a vesicle, is exocytosis. And you can see a picture here of these are the cells that form, they're called the endothelium. They're the cells that form blood capillaries. So basically the green molecule we can see on the left is being brought into the cell, and it's being brought in by like the plasma membrane sort of pinches off around that solution. Excuse me. And when it pinches off, it moves that solution into the cell. And that is going to remove a certain amount of the plasma membrane, because that plasma membrane pinches off and forms the vesicle. And then if the cell wants to get rid of that substance, in other words, this is showing you the movement of material from one side of a cell all the way to the other side of a cell, so all the way across that cell. And then that vesicle fuses with the plasma membrane and ejects the material. So now the plasma membrane is going to get bigger. So it got smaller during penocytosis by pinching off and forming a vesicle, and it got larger when a vesicle fused with the plasma membrane. So the amount of endocytosis and exocytosis needs to be balanced so that the plasma membrane remains the same size. Now, we also have, in addition to phagocytosis and penocytosis, we have receptor-mediated endocytosis. And that is just endocytosis of substances, solute particles, molecules that attach to receptors on the plasma membrane. And when the vesicle pinches off, those receptors coat the membrane of the vesicle, and those receptors have the molecules attached inside the vesicle. So in other words, this is a specific process. It is specific to, for example, one molecule that enters the cell this way by receptor-mediated endocytosis is LDL, which is low-density lipoprotein, which is what we know as bad cholesterol. So bad cholesterol enters our cells by binding to receptors and then entering by receptor-mediated endocytosis. This is just showing you exocytosis. Now we're gonna talk about the parts of the cell. So the cytoplasm and the cytosol are closely related, but they're not identical. The cytoplasm is everything within the space between the nucleus and the plasma membrane. So it is cellular material outside the nucleus, but inside the plasma membrane. That means it includes the fluid inside the cell, but it also includes the cell parts called organelles. So the cytoplasm is actually composed of the fluid portion, which is cytosol, the protein fibers, which form the cytoskeleton, the cytoplasmic inclusions, which are just very large molecules, but they do not have a membrane around them. Pigment molecules inside of a plant cell are inclusions. And then the organelles, or little organs like the mitochondria, that play certain roles in the maintaining the cell. The fluid portion of the cytoplasm is the cytosol. Okay. The cytoskeleton consists of 3 protein fibers. Microtubules are the thickest of the fibers, and they're made of a protein called tubulin. We find microtubules they form these spindle fibers during cell division. They are components of centrioles, which are important during cell division. They are components of the cilia and flagella, and they play a role in the movement of cilia and flagella. And cilia are little hairs. Flagella is one tail. You have flagella in sperm, and then cilia line our respiratory tract and help sweep mucus up and out of our lungs so we don't get lung infections. So the cytoskeleton, it's not bones. It means the skeleton of the cell. Okay, cyto means cell. So the cytoskeleton supports the cell but allows for movements like changes in cell shape and movements of cilia across the surface of the cell. Microtubules are the thickest of the cytoskeleton fibers, and they do form an internal scaffold, and they're involved in transport and cell division. Microtubules are components of centrioles, which produce centrioles arrange the spindle fibers during cell division. And the spindle fibers, their role is to attach to the chromosomes and pull them apart when the cell divides. And then you have microtubules in cilia and flagella. Then we have the thinnest. So microtubules are the thickest. The thinnest filaments are called actin filaments, and they are thin, or known as microfilaments, because they're so small. So we have microtubules, or thick filaments, and then we have microfilaments called actin filaments. And we're going to see actin in cell structure. They're going to support the microvilli, those folds on the surface of certain cells that increase the surface area. Actin is going to play a role in the contraction and movement of certain cells. And then there are intermediate filaments, which are the medium thickness, and they provide the mechanical strength of nerve cell extensions, like the axon fibers. Then we have cytoplasmic inclusions, which are aggregates or collections of chemicals such as lipid droplets, melanin, which is a pigment, or the brown pigment in our skin, glycogen, which is a polysaccharide that the liver takes up glucose and stores it in liver cells as glycogen. And then lipochromes, which are, it's a a lipid attached to another molecule because that's the only way it can be transported in the body fluid. Okay, now the organelles, they are small specialized structures with particular functions. We'll start with the nucleus, which is the largest of the organelles. It is a large membrane-bound structure, and the important thing about the nucleus, it contains the DNA, and of course DNA codes for our traits. Excuse me. DNA is located in the nucleus and codes for our traits. It codes for specific proteins. And then since we're made of protein, that's why we say that DNA codes for our traits, because it ends up determining our genetic traits, our genetic characteristics. RNA assists DNA in making protein. And then there's a structure inside the nucleus called the nucleolus, and the nucleolus manufactures ribosomes. So the nucleolus manufactures ribosomes. Ribosomes make or synthesize proteins. And then the nucleus itself contains DNA, and DNA and RNA are just, DNA codes for RNA, and RNA is going to code for the protein. It's going to be translated into the protein. As far as chromosome structure, during much of the cell cycle, chromosomes are dispersed as chromatin. Chromatin is very diffuse, spread out DNA and wound around histone proteins. So chromosomes are made of DNA plus proteins. So DNA is found in the nucleus associated with proteins. In other words, it winds itself around histone proteins, as you see here, and forms nucleosomes. Nucleosome is just this collection of DNA wound around a histone or a collection of histones. So the DNA plus the histones form the chromosomes. But much of the time when during the life of the cell, chromosomes are dispersed as chromatin. And then when the cell is about to divide, during cell division, the chromatin condenses and it compacts into chromosomes. So the chromatin is all spread out and you can't see it very clearly under the microscope. But when the cell begins to divide, the chromatin condenses and packs itself tightly into these rod-shaped structures called chromosomes, and those you can see under the microscope. In fact, that's how we do a test and produce a picture called a karyotype that shows all of our chromosomes, and you can do this to determine, for example, if a child has a chromosomal abnormality like Down syndrome. Ribosomes, remember, are manufactured by the nucleolus, and ribosomes synthesize proteins. Ribosomes can just be free in the cytosol or attached to the endoplasmic reticulum, forming the rough endoplasmic reticulum. The rough ER and the smooth ER. The ER stands for endoplasmic reticulum, and rough ER has attached ribosomes, so therefore rough ER manufactures proteins and modifies proteins. The smooth ER does not have attached ribosomes, so it's smooth. And it's going to manufacture lipids. I always remember this because lipids are fats and oils, and if you feel of fats and oils like in your fingers, they're going to feel smooth. So I always remember it that way. The smooth ER also participates in detoxification, so liver cells have quite a bit of smooth ER in them, and muscle cells have smooth ER that stores calcium ions because they're needed for muscle contractions. I guess I'll stop here because I've already gone to 20 minutes. So we'll start back at slide 59 and we'll do our last recording, starting with slide 59 to the end of the slides.